Well, it hasn't really been that long, but science waits for no one, so there's even more to tell. Hello and welcome to another episode of You and Me and Multiple Sclerosis. My name is Pam and I've been living with multiple sclerosis for almost 40 years now. Lately, there's been some interesting research and advances on remyelination, which we all know is the thing that we are looking for. If you've had MS for any length of time, you have damage from demyelination, the loss of the myelin sheath around your nerves, that accounts for a lot of your symptoms and problems. But it doesn't go away, especially if you're in the progressive stages. The damage just accumulates over time. And slowly, or maybe not so slowly, we get worse. So we're all looking to remyelination to fix that for us, to turn things around. But how close are we? Well, in the videos that I've been putting out recently, it hasn't really sounded like we're ready for prime time, though important advances are being made. I thought that by putting one up a couple of weeks ago, I was going to be able to put that topic aside for a while. But now I'm seeing new articles that are coming out. So I thought that without further delay, I would go ahead and keep you up to date on what I'm seeing. I'm not going to do a lot of commentary because I don't want to make this video very long. I'm going to go ahead and link below the video that I did recently on remyelination, so you can watch that one too. And that will hopefully give you the full picture of what's going on, or at least as far as I know so far, what's going on with remyelination. So let's go ahead now. We'll turn to those articles and take a look. The articles we'll look at today are from Multiple Sclerosis News Today, and I will look at them in the order they appeared. They're both in May, so they're very recent. Discovery may ultimately help to repair myelin in brain. Mouse study. Scientists find way to turn brain cells into myelin-repairing oligodendrocytes. And you can see here that it was published on May 21st of this year, 2024. And they say that scientists in Iran have discovered a new way to convert astrocytes, star-shaped brain cells that support nerve function, into oligodendrocytes, the cells that make and repair myelin in the brain. That's an interesting one. So converting one kind of cell into another kind of cell. When the converted cells were transplanted into the brains of mice with myelin damage like that seen in multiple sclerosis, the converted cells were able to help repair damaged myelin. The generation of OPCs, or oligodendrocyte progenitor cells, from astrocytes via a small molecule cocktail may provide a new avenue for producing required progenitors necessary for myelin repair in diseases characterized by the loss of myelin, such as multiple sclerosis, the researchers wrote. The team described their new method in a study titled Conversion of Astrocyte Cell Lines to Oligodendrocyte Progenitor Cells using Small Molecules and Transplantation to Animal Model of Multiple Sclerosis, which was published in the Journal of Molecular Neuroscience. Given that astrocytes are abundant in the brain and spinal cord, they are an attractive source for generating OPCs in demyelinating conditions, the researchers wrote. And they say that they're seeking ways to grow cells that could help with myelin repair in the brain. In MS, inflammation in the brain causes damage to the myelin sheath, etc., etc. I'm not going to read that because we all know what that is. While neurons are the most well-known type of brain cell, astrocytes are about 40% more abundant than neurons in the human brain. These cells have a wide array of important functions, helping to regulate metabolism and inflammation in the brain, as well as supporting neuron function. Astrocytes also are able to shapeshift. Under certain conditions, these cells can grow and differentiate into oligodendrocyte progenitor cells, known simply as OPCs, which basically are immature oligodendrocytes. So I guess this astrocyte is the type of cell that can differentiate. This is something that they're going to take advantage of then to get it to differentiate into the thing that they want it to differentiate into. And they say that in this study, scientists at the Tarbiyat Modares University in Tehran reported a novel method to grow astrocytes into OPCs in a lab setting. The method requires a unique cocktail of small molecules that are added at specific concentrations to the medium used to grow the astrocytes. Other than its creation, the method is pretty straightforward. The cells grow in a dish, and all the researchers need to do is refresh the medium every few days. By the way, just as an aside, the study is coming out of Iran. I have been noticing a lot more clinical trials and studies coming out of Tehran. And I've also been reading that the number of MS cases is growing exponentially in Iran. The scientists showed that OPCs grown by this method exhibited all the major biochemical characteristics typical of this cell type. Most importantly, they were able to grow into mature myelin-making oligodendrocytes. Carrying their work further, the team then tested the effects of transplanting the astrocyte-derived OPCs into the brains of mice with MS-like disease. In this model, animals are fed a toxic chemical called cuprazone that causes MS-like damage to the myelin sheath. Consistent with results seen in dishes, the transplanted OPCs were able to grow into mature myelin-making oligodendrocytes in the mouse model. 2 weeks after transplantation, most of the grafted cells expressed markers of mature oligodendrocytes, the researchers wrote. The team said that this finding is remarkable considering that OPCs tend to have difficulty growing into mature oligodendrocytes in the cuprazone-induced model. Given these data, the scientists suggested that OPCs created by this method could be a useful basis for the development of cell therapies for diseases like MS that are characterized by myelin damage. Helping the strategy is that astrocytes are abundant in the brain. Yeah, that is good news. Because even if a portion of them differentiated the way we wanted, it would still be a significant number. And then they say developing efficient and safe methods for generating OPCs is a promising strategy for treating demyelinating diseases, including MS, the scientists wrote. And I guess I will take another moment here just to give thanks to those little mice who, even though it wasn't really their choice, have been giving their lives for our benefit and for hopefully down the road a cure for multiple sclerosis. And then there was this second article, which was published on May 24th of 2024. Progentos receives $65 million to develop myelin regeneration therapies. The company is developing small molecules to induce remyelination. Sounds promising. Pento Therapeutics said it received $65 million in funding to support the development of myelin regeneration medications able to restore function in people with multiple sclerosis and other diseases marked by myelin loss. The biotech company said it will use the funds to advance its MS program to proof-of-concept clinical studies and expand its myelin-restoring small molecule pipeline to other degenerative conditions. While there are many treatments that are highly effective at slowing the progression of disease, there is significant unmet need for new approaches that can regenerate myelin and restore function for patients with MS, Chris Luce, PhD, Progentos CEO, said in a company press release. Luce founded the company along with Sanjay Maghavi, PhD, Chief Scientific Officer. MS is caused by a Okay, we'll skip that paragraph because we already know all that. We'll go here. Although available therapies can reduce immune attacks to the brain and spinal cord and slow disease progression, they are not able to repair damage that has already occurred. That means there's an unmet need for remyelinating or myelin repairing therapies that can potentially restore some of the lost function. They say that small molecules stimulate the body to repair myelin. Progentos is developing small molecules designed to induce remyelination by increasing the number of oligodendrocytes, specialized cells that produce myelin, in the brain and spinal cord. The therapies work. Doesn't this sound pretty similar to what we just read? That we're going to increase the number of oligodendrocytes. And let's see if they talk about the mechanism, if it's astrocytes or something else. The therapies work by promoting the differentiation of oligodendrocyte progenitor cells into fully mature oligodendrocytes, an approach that essentially stimulates the body's natural repair mechanisms to replace the lost myelin. This does sound like the other one, doesn't it? But I haven't seen them say anything about astrocytes yet. According to the company, scientists have already identified and validated a novel target that regenerates myelin in animal models. Progentos' proprietary molecules, so we're not going to tell you what they are, have outperformed previous approaches to differentiating oligodendrocyte progenitor cells into new oligodendrocytes. I am thrilled with the support received from this group of investors and appreciate their recognition of the need to advance the current standard of care for the millions of individuals impacted by MS and other demyelinating diseases, Loos said. The financing round was led by Netherlands-based Forbion and joined by Alta Partners, Mission Biocapital, Longwood Fund, and Dolby Family Ventures. We are truly excited to be part of Progentos' journey towards delivering potentially disease-modifying drugs for diseases like multiple sclerosis, diseases with high unmet clinical needs, said Dmitry Hristodorov, PhD, general partner at Forbion. Chris and Sanjay are bringing a high level of technical expertise, entrepreneurship, and leadership, and we are honored to support them in this endeavor. So while this is not the same as the one we just read, it does look like they're both looking at the same thing, that we need to be able to turn these progenitor cells into actual oligodendrocytes, and that will then help stimulate the production of myelin. Very, very promising. And here's one that I see was linked from the article we just looked at. It was published January 31st of 2024, but somehow I don't think I saw it before. It's called Myelin Repair Early in MS May Protect Against Disability Worsening. The approach may be most effective in preventing long-term clinical progression. Well, that's kind of exciting, I would say. They say that people in the early stages of multiple sclerosis who have more spontaneous myelin repair in the cortex, the outermost layer of the brain that's critical for higher cognitive abilities, are less likely to experience worsening disability. A new analysis suggests these findings have important implications for how therapies to promote remyelination might best be used, according to researchers. A combination of available disease-modifying therapies for MS that reduce inflammation and myelin damage and emerging therapies that promote myelin repair or remyelination early in the course of the disease may be most effective in preventing long-term clinical progression in MS, they wrote. So the sooner the better. The more damage you accumulate, the harder it is to get it all repaired. So that only makes sense to me. They say the study, Time is Myelin, Early Cortical Myelin Repair Prevents Atrophy and Clinical Progression in Multiple Sclerosis, was published in Brain. Sounds like an interesting one. The articles will be linked below and you can click on these yourself, but that one might be one worth following up because it doesn't even sound like it's written over my head, at least from the title. The myelin sheath is a fatty substance that wraps around nerve fibers and helps them send electrical signals. In MS, the immune system attacks myelin, which disrupts nerve signaling and eventually causes nerve cells to sicken and die, leading to MS symptoms and driving long-term disability. Some remyelination occurs naturally in MS. Although this general mechanism is pretty well established, the exact timing and dynamics aren't. It's also known that there is some remyelination that occurs naturally in MS, but how this connects with long-term disability outcomes isn't clear, and few studies have looked at remyelination in the cortex specifically. In this study, researchers analyzed MRI and clinical data from 140 people spanning all types of MS who were followed for at least 5 years at centers across Europe. I like that there's a little bit more geographic distribution than some clinical trials tend to be, and also a goodly number of people, and I like how they included all types of MS. And 5 years, that's very good. So there's some really positive aspects to this study. That means that the results should actually be meaningful for us. They say that of note, several of the researchers have financial links to pharmaceutical companies. And of course they do. Because, it's all about making some money at the other end. And I guess I'm not going to begrudge them the money. If they can fix my MS, I'll let them make money on that. And then we'll all be richer. All of the patients underwent specialized MRI scans to detect myelin content twice, about a year apart. From these data, the researchers calculated 3 key variables: the total amount of demyelination or myelin loss present at the first scan, or baseline, compared with a control group of 84 healthy individuals of the same age and sex, and the amount of demyelination and remyelination between scans. The researchers noted there was a lot of variability in these measurements, but said spontaneous remyelination was extensive in half of patients. There also tended to be more myelin loss and less myelin repair at the very edge of the cortex compared with layers deeper in. The outer band of the cortical ribbon displayed a more pronounced extent of demyelination and a lower extent of remyelination compared with the rest of the cortex, the researchers wrote. None of the myelin measurements showed clear associations with patients' age or MS type, which is interesting. Individuals who'd been living with MS for longer have significantly more extensive demyelination at baseline and over time, but the amount of spontaneous remyelination showed no association with disease duration. Well, this is excellent news if that's going to turn out to be true, because all along we've been thinking those of us who've had more years to accumulate demyelination damage are least likely to benefit from remyelination. Importantly, our data demonstrate that the extent of cortical remyelination in people with MS is not influenced by chronological age, disease duration, or clinical phenotype, the scientists wrote. They also noted patients who had more demyelination and less myelin repair over time experienced significantly more atrophy or shrinkage in the cortex, which is consistent with the idea that myelin damage leads nerve cells to sicken and die. 40 patients experienced significant disability at 5 years. Over the course of follow-up, 40 of the patients had experienced clinically significant disability at 5 years. The researchers constructed statistical models looking for significant connections between measures of myelin in the cortex and the risk of disability progression. Results showed that patients with more extensive myelin loss at baseline were significantly more likely to experience a worsening of disability. The models also showed a significant interaction between remyelination, baseline demyelination, and the amount of time patients had been living with MS. Okay, is that saying something different from what they just said a minute ago? The models also showed a significant interaction, so a relationship, between remyelination, baseline demyelination, so how much damage had already accrued, and the amount of time patients have been living with MS. I'm a little bit puzzled about that because I thought they just said earlier that there wasn't any connection. In other words, here's the explanation. Let's hope. In other words, a greater extent of cortical remyelination was associated with a lower probability of clinical progression at 5 years, only in the subgroup of people with MS with a limited amount of cortical demyelination at baseline and a shorter disease duration, the researchers wrote. Okay, now that you've explained it, I still feel like you said something kind of the opposite of what you said earlier. But maybe I'm just not understanding it. Well, my husband is a scientist, so I asked him, and he read through it, and he said, yeah, it does seem to be saying contradictory things. So I feel better. I'm not imagining it. Based on these findings, the scientist said, if treatments to reduce demyelination and promote remyelination could be given early in the course of MS, it could have a transformative impact on the evolution of the disease in the long term by effectively preventing neurodegeneration and its irreversible clinical consequences. Well, that word irreversible also kind of puts a little bit of a damper on the whole excitement that I started to feel there for a minute. Because, yeah, I guess at some point damage is still irreversible, even with this new therapy. So, all right, once again, guardedly optimistic, but I'm not really sure I understand what it is that they are really saying here. I'm going to have to study this a little more. Okay, I think I'm going to jump in here and interrupt myself. I recorded this video earlier in the day, and then later the same morning, I had an online support group meeting as part of the clinical trial at OHSU that I've been on. And the support group administrator said something very interesting. She said that, according to what she understood, those of us who were participating in this clinical trial would be invited to participate in the next one that they're going to do. And I was quite excited to hear about it, because this one involves a drug for remyelination. Now, I have no idea yet whether I'll even qualify for it, but I'm excited to know that they're working on it. And right here in my own backyard, practically. Well, before I get too excited, though, I just better remind myself what Professor G has said many times. And I've shared this with you before, but it's always good to be reminded, because he is actually optimistic that a cure will be found. So he's not one of the doomsayers. And yet, even so, his tone is kind of cautionary.
So, I mean, the holy grail is taking somebody who's got established MS and giving them a treatment to, recover their function. And it's based on the hypothesis that people with MS have demyelinated axons, and you can repair those axons by remyelinating them. I think that's kind of naive, because, people with more advanced disease, when they've lost their axons, you can't remyelinate them.
Well, that's okay, because we're going to continue to be hopeful, despite the fact that, yes, it's true. Those of us who are older than 55 and have had damage that's gone on for a long time probably can't expect to see a lot of improvement, even if they do come out with something that will remyelinate nerves. Even so, I'm excited about the progress, and I hope you are too. Well, as I said, I'm not going to add a lot of additional commentary. I talked a little bit throughout looking at the articles when I saw something that piqued my interest or that I had some observation to make. But other than what I already said in my prior video, I'm just going to reiterate that I think we just need to be patient and wait, but at least this gives us some hope that they're moving in a good direction. Because that's what we're all waiting for, is the day when our doctor says to us, guess what? We have remyelination therapy now. Would you like to try? I don't know how long that will be. But in the meantime, let's just take really good care of ourselves so when the time comes, we are ready. And I'll see you again in my next video.